Hi everyone, welcome back to the GEP Pathways video walkthrough. My name is Kirsten. I'm one of the GEP virtual TAs with this program, and today we will continue on to part 6. Now that we've mapped each CDS separately to determine their approximate locations, we will further refine the CDS boundaries by searching for compatible splice donor and splice acceptor sites by visual inspection using the genome browser. This diagram shows us what the process of translation looks like, going from pre mRNA to processed mRNA. Before translation of our processed mRNA occurs, the introns in our pre mRNA are spliced out, hence why we refer to our GT and our AG sequences as our splice sites. Our start codon, ATG, which codes for methionine, will signal translation to begin for our coding sequence. As the ribosome goes along the coding sequences of our processed mRNA, it will encounter a stop codon at the end. This stop codon signals a termination of translation, and our end result will be a functional coding protein. As part of the ModEncode project, RNA-seq data for D. yacuba was generated using samples from adult females and males. These RNA-seq reads, usually 100 to 125 base pairs in length, are derived primarily from processed mRNA, which is after the introns have been removed. Hence, our genomic regions with RNA-seq read coverage usually correspond to transcribed exons, which include both the translated and untranslated regions. The RNA-seq tracks correspond to the samples from adult females, which is in red, and males, which is in blue, where our RNA-seq data is available. The height of the histograms within each track corresponds to the number of RNA-seq reads that have been mapped to each position of the D. yacuba scaffold. By default, the scale of the RNA-seq coverage track will change automatically based on the minimum and maximum read depth within the genomic region being viewed. We need to ascertain whether the T-blast N alignment for CDS1 at 19,150,809 to 19,150,856 is supported by RNA-seq data, and if so, determine the location of the start codon. So let's return to the genome browser for D. yacuba. To examine the region of the T-blast-N alignment for CDS1, enter NC underscore 052530 colon 19150809 dash 19150856 into the enter position or search terms text box and then click go. If we scroll down under the mapping and sequencing tracks, change the base position track to full and then click on any refresh button. The RNA-seq tracks for both samples show high RNA-seq read depth within the T-blast-N alignment block, consistent with the hypothesis that this region is being transcribed in D. yacuba. Notice that frame +1 of this region has two stop codons, which is denoted in red with an asterisk, while frames +2 and +3 have an open reading frame, meaning that there are no stop codons shown within frames +2 or +3 of CDS1. To examine the region surrounding the start of the T-blast-N alignment to CDS1, enter NC underscore 052530 colon 19150809 into the chromosome range or search terms text box. Then click on the Go button and zoom out 3 times and then another 10 times. In part 5, we found our T-blast-N alignment for CDS1 begins at approximately 19150809. Examination of this region using the genome browser shows us that frame +3 has a start codon in this location and is supported by multiple evidence tracks such as the NCBI RefSeq genes and spawn alignment of D. melanogaster proteins, and our gene predictors such as Gemoma, NSCAN, and Augustus. The T-blast-N alignment for CDS1 of Reb and D. melanogaster against the D. yacuba scaffold encompasses all 16 amino acids of the CDS, and the alignment begins with a start codon at 19150809 to 19150811. Hence, the most parsimonious gene model for Reb PA in D. yacuba would use the start codon at 19150809 to 19150811 in reading frame +3 in our scaffold NC underscore 052530.